Good morning, everyone. Thank you for joining us and welcome to our webinar today, qPCR 101, What, When and How. We'll shortly commence with the webinar, but before that, we have a couple of housekeeping slides to get through, so bear with us for a minute. So housekeeping, today this webinar will be recorded. It will be available to you at a later date and you'll be contacted with the web links for the recording. You are all on mute and video off. So if you do have a question today, please type it into your Q&A panel on your screen, and we'll endeavour to get to those questions either throughout the talk, we may have some time, or definitely at the end of the talk, we will have a Q&A session. Lastly, also, before you leave at the end of the talk, there will be a post-event survey that you could attend. Please, your feedback is always welcome and valuable. It helps us make these webinars bigger and better for you all. Next slide. So to our speaker today is Dr. Bernice Tan. She's your local field application specialist, and by local I mean she's based in Melbourne. She's the FAS here at Merck for genomics and cell culture, and you'll be hearing her throughout the course of this webinar. You'll have two moderators. One is Dr. Yi-Cheng Kuo. He's the Merck marketing specialist in this space. And lastly, me, I'm John Roniotis, and I'm the field application specialist manager. And just as we wait for the next slide. Okay, so this is the second webinar we have in this series. A fortnight ago, we had the Winning Western seminar. Today, again, we got qPCR 101, and we'll have 3 more seminars in this series coming to you. You would have received, hopefully already, an invitation to the next one, which is a bit different, which isn't anything qPCR-based. Peaky Blinders, tips for improving LC-MS sensitivity. And in the coming weeks, we'll have a sample prep and filtration seminar, and close off again by Bernice, a 3D cell culture, the new Lego seminar, which will be our last one for this series. So, without further ado, enough from me. Take a seat, have yourself a warm cup of coffee, and I'll throw over to Bernice, who will carry you through for the rest of this morning. Thank you.
Thank you, John, for the introduction, and hello, everyone. Thank you for your time this morning. So today we're going to talk about what qPCR 101, what, when, and how. We'll touch on the best practices in order for you to get qPCR data that you can be confident with. Just a little bit about myself. John mentioned I'm based in Melbourne. I came from the research lab, graduated from the PhD from the Garvan Institute, looking at TNF-alpha signaling in the context of type 1 diabetes. During my PhD, I had extensive wet lab experience with in vivo mouse models, cell culture, as well as a number of molecular biology techniques, including PCR, qPCR, and droplet digital PCR. I've been in the industry for 8 years now, 2 years with Merck. I've spent a lot of time working with customers like yourself, troubleshooting your PCR molecular techniques, as well as helping some of the labs actually set up their PCR techniques. So just getting warmed up. So on the right-hand side of your Blue Jeans app, you will see above the Q&A, a poll is currently switched on. So we're just going to test this out. So the first poll, so we have 3 polls in this webinar today. The first poll is, do you currently do qPCR? I'll give you 10 seconds to pop your questions in, or your answers in. Thank you. Okay, let's review the answer. Everyone found the poll button on your right?
Bernice, we may have had a slight malfunction with that poll, but it was at 50% was I'm new to this, and 30-something% was I occasionally do qPCR.
Okay, that's good. Thank you, John. I might need you to review the answers if I can't see them. Okay, so we'll move on. So that's good. I hope that everyone will get something to take home with you today in terms of our qPCR 101. So what we're going to talk about today is for those of you who are new to qPCR, we'll touch on what is qPCR and when it can be used, so the types of application. We will then spend majority of this presentation on how do you set one up, how do you set up your qPCR assays, and how do you validate your runs. And then based on the information that I've collected in the registration information, a lot of you have listed some of your common qPCR issues. So we've picked out 2 of the most common ones to discuss. And if you've got any other questions, please pop them in the Q&A. We'll do our best to answer them today. If not, we'll get in touch with you to help you out. All right, so let's just get started. Okay, so an overview. What is in a PCR reaction? So PCR stands for polymerase chain reaction. It consists of 4 main components. So you need a template for your PCR. It can be DNA or RNA. If you're starting with RNA, you do need to convert it into cDNA by a process called reverse transcription. So we'll talk more about reverse transcription in a few slides down. The next thing you'll need in a PCR reaction is primers, forward and reverse, to pick up your product of interest. You also need an enzyme, DNA polymerase, as well as buffer components in your PCR reaction, which includes DNTPs and cations such as magnesium chloride. The 2 components here, the enzyme and the buffer, are available in different formats. You can get them as single tubes, so each component in a single tube. That gives you the flexibility to optimize individual components for your PCR. But a lot of people also choose to go with a single optimized tube format. At Merck, we call this the ready mix. So what you do in a qPCR or a PCR is you add these 4 components in a tube, a PCR tube, and then you pop it into a thermocycler or a PCR machine. And you cycle them through your PCR reaction through these 3 main stages. So you've got the denaturation step. Depending on your enzyme, it's usually at 94 to 95 degrees. So you heat up your tube to these temperatures to allow your double-stranded DNA that's really, really tightly bound together to unwind. So this provides accessibility for your primers to bind at the next stage. So when you cycle through to the annealing stage, this usually occurs somewhere between 55 to 65 degrees, depending on the primer set that you have. This is the temperature that allows the primer to bind to where you've designed the primers to bind to within your template. And then you increase the temperature to 72 degrees, and this is where the polymerase incorporates DNTP to lengthen out your primer. And this in light blue here is your first set of PCR product. So PCR is used to amplify DNA. Okay, so you cycle through this 2nd and 3rd step multiple times until you get more and more of these PCR products to enable you to analyze them. So in a traditional PCR, you would load your products on a gel to visualize them. So it tells you presence or absence. You usually have a ladder to tell you, to enable you to detect or find out if the amplicon size that you're after is present. So in this case, this particular product here or PCR product here is 150 base pairs. Now qPCR or real-time PCR is built from traditional PCR. Real-time PCR, or otherwise known as qPCR, quantitative PCR, is basically traditional PCR, but there's an optical module added to the PCR machine, and a fluorophore is added to the PCR reaction. So this allows you to view the reaction in real time. So usually with your real-time or qPCR, you will get a qPCR amplification curve that is sigmoidal in shape, looks like this, and it consists of an initiation phase, followed by an exponential phase. This is where your PCR products double with every cycle. And then eventually it stops and it plateaus off. So the exponential phase is where you quantify DNA. You do this by drawing a threshold at the lower 1/3 of the exponential phase of the curve, or you can get your qPCR machine to calculate this automatically. But basically, where the threshold crosses the amplification curve is where you derive a CT or quantification cycle. And this is how DNA is quantified. So why would you perform qPCR? It's there for you to quantify expression of genes or DNA sequence of interest within your sample. So what is it commonly used for, or when do you use qPCR? So the most common application, I think for most of you, will probably be gene expression analysis. But qPCR can also be used for things like NGS library quantification, validation of RNA-seq data, and microarray data. If you're doing functional genomics, qPCR can be used to validate knockdown or knockout by RNAi method or CRISPR-Cas9 systems. qPCR is also commonly used for genetic variation analysis, such as copy number variation and SNP detection. qPCR is also a really important method used in diagnostic and applied applications, which a lot of you today are also doing. So for example, it's used for pathogen detection, so virus and microbes, and in this current pandemic, qPCR is the main method used for detection of COVID-19 virus. qPCR is also there to check for food spoilage or water pathogens in the water and environmental samples, so it helps keep our food and water safe. But less common, but it's also used in chip analysis as well as protein analysis. I think the main point here I want to list is that it can support many applications. I mentioned before that if you're starting with RNA instead of DNA, you need to first convert the RNA into cDNA by a process called reverse transcription in order for qPCR detection. So this can be done using a process called RT-qPCR. So RT-qPCR stands for reverse transcription quantitative PCR. So basically it's a technique to allow you to amplify messenger RNA, where cDNA is created from mRNA, and then that cDNA is used as a template for qPCR detection. Now, I just want to highlight that there's two types of RT-qPCR you might have here at some point. There's one-step and two-step. So one-step is where RT and PCR occurs in one tube. So this is the method of choice for many diagnostic applications. The reason being there's less handling, so there's less risk for contamination. It's also easy and fast to run. A lot of research people or researchers tend to use two-step RT-PCR. This is where RT and PCR occurs separately in different tubes. The reason for this, the choice for this particular method is because it allows you to optimize each step. It also allows you to load more cDNA to increase sensitivity of your assay. You can also then analyze your cDNA to look for multiple targets. So it allows you to stretch your results further. Now you know what qPCR is and what RT-PCR is. We can move on to best practices for qPCR. Now, before I get started, the whole basis of best practices of qPCR basically comes from MyKey Guidelines. For those of you who are new and those of you who are already doing qPCR, I suggest that if you don't know the MyKey Guidelines, you should read more about it here. So you can visit this link here for more information. So MyKey stands for Minimum Information for Publication of qPCR Experiments. The reason for these guidelines is this group of thought leaders, so they're actually really well known in the world of qPCR. They got together and they realized that there's actually lack of consensus on how people are publishing qPCR information. So that leads to a lot of frustration in the field. People didn't know what to do and how to tackle qPCR obstacles. So they put together these guidelines to address these challenges. So these guidelines are there to promote experimental transparency between labs. It's there to maintain integrity of scientific literature, and it allows you to publish and bring your product to market faster and with confidence. So if you look at the MyKey guidelines, there's actually a lot of components of qPCR that you do need to consider. But in the interest of time, and for a lot of you who are new, I thought I'll put together these 3 main key considerations when setting up your qPCR. So what we're going to touch on today is sample preparation, your qPCR chemistry and assay design, as well as your qPCR assay validation. So we'll start off with sample preparation. I like to start off by saying that your analysis is only as good as your sample. And I think this same theory applies to any sort of experiments that you do in the lab. So if you start off with not so good sample, you're not going to get good results. So always remember, garbage in, garbage out. So make sure you start off with good samples. And how do you do that? Okay, so the first step to good sample preparation consideration is sample storage and using an appropriate extraction kit. So I want you to take home 3 main messages here. So when you think about sample prep, think about how you store your sample. So if you collect a piece of tissue, plant sample, or blood, if you're not able to extract the sample straight away, make sure you stabilize the nucleic acid. So you can use a nucleic acid stabilization reagent, such as RNA later for tissues, or you can snap freeze your sample in liquid nitrogen if you're allowed to handle liquid nitrogen in the lab. So look for a method that fits your sample type. You also need to think about selecting an appropriate kit for your sample and nucleic acid type. So there are many different types of samples out there. Some are easier to extract, such as cells, compared to things like tissue and plants and stool. So make sure you select the right kit. So we provide different specific kits for specific samples because it allows you to handle the samples a lot better. It provides it has extra steps to allow you to assess or provide accessibility to the nucleic acid. And also some samples such as stool and blood plasma tend to have a lot more inhibitors. So the extraction process actually helps you clean up some of these inhibitors to give you the best results. Now you also need to think about whether you're extracting DNA or RNA. And with RNA, whether you're looking at total RNA or you want to enrich for small RNAs. So there's specific kits for specific applications. Once you extract your DNA and RNA, make sure you store them properly as well. So you want to store them in -20 or in -80 for long-term storage. And my recommendation for you is to make aliquots to avoid multiple freeze thaw. So maybe try not to freeze thaw them more than 3 times because freeze thawing cycles can actually cause your nucleic acid to degrade. So this takes us to the second poll, which is currently switched on. So the question I have for you is, how do you check for purity and integrity of your RNA? So this is really important. Doesn't matter whether you're extracting RNA or DNA, you need to think about QC of your sample. I'll give you 10 seconds to answer this question. So let's have a look at the results. Okay, so a lot of you are saying here that you are using a spectrophotometer, such as a nano drop. And then some of you are using a spectrophotometer, such as nano drop for purity, and then also running an RNA gel or using the bioanalyzer. I'm really happy to do that. So thank you for your time. So let's move on. Okay, so once you extract your nucleic acid, it's really important to quality control them. So I'd like to highlight the fact that when you QC your nucleic acid, you need to think about purity and integrity. A lot of researchers confuse between these two. So purity is not the same as integrity. When you talk about the purity, you are measuring the purity of the sample using spectrophotometer. You can use a standard spectrophotometer for this application. It can take a little bit more sample than usual. So a lot of researchers use a nano drop for assessing the purity of your sample. So the nice thing about the nano drop is it only requires 1 to 2 microliter of your sample. So when you look at the purity of your sample, you are basically looking at the ratio, the absorbance ratio of A260 and 280, as well as the absorbance ratio of A260 over A230. You want both of these values to be greater than 1.8. So A260 is the absorbance for pure RNA, and A280 is the absorbance for protein contamination. So if your A260 over 280 value is a bit low, I would recommend, so probably you have protein contamination, I recommend cleaning up your sample. Same for your A260 over 230. If it's a bit low, it indicates that there are other contaminants that are possibly carried over during your extraction method. It's really important to clean up your sample. You can do that by running it on a column if your kit allows that. So check your kit manual, or otherwise you can perform an ethanol precipitation to clean out impurities. Impurities can inhibit your downstream RT-qPCR, so it's really important to check and clean up your sample. Now you've got good pure samples. You need to also look at integrity of your DNA and RNA. We're talking about RNA here. So in terms of RNA, you can assess integrity by looking at the ribosomal subunit bands. In eukaryotes, there is the 28S and 18S subunits, and in bacteria, 23 and 16S units. You can do this by either running an agarose gel, 1%, that requires quite a bit of sample. So a lot of researchers like to use a microfluidic analyzer such as the tape station or bioanalyzer. The beauty of this system is they only require 1 to 2 microliters of your RNA sample, gives you quantity and quality of your RNA. It spits out a number. So with the bioanalyzer, you get a RIN or RNA integrity number. So it allows standardized reading for all your samples. And these microfluidic analyzers are usually available in core facilities, and you can use it for a small fee. So if you run it on a gel, so these are eukaryotic RNA samples, you should see 2 distinct bands, 2 nice sharp bands for the 28S and 18S ribosomal subunits. Now, if you can see here in lane 3 and 4, if you run your RNA sample and you see a smear like this is an indication that your RNA has degraded. So you do need to start again, go back to your sample prep and sample storage and handling steps. Think about how why your samples might have caused degradation, and then start again, and hopefully you'll get good quality samples for analysis. Now you've learned how to extract and QC your nucleic acid. Now we're going to talk about qPCR chemistry and assay design consideration. There are 2 main qPCR chemistries, the intercalating dye, cybergreen, or hydrolysis probes. Some people call these TechMan probes as well. So in terms of cybergreen assay, it's a dye that binds to all double-stranded DNA. So it's generally seen as low specificity, but the beauty of it is it gives you increased sensitivity, and you don't need any probes. So the forward and reverse primers are quite cheap to purchase, and they generally are delivered to you within a week. Now the disadvantage is it can pick up non-specific products, but you can check this with your cybergreen assays by running a melt curve analysis, as well as running your products on an agarose gel post-PCR. We'll talk more about these later on. Now with the probe-based detection, it's a general method of choice for critical applications such as diagnostic applications. The main reason is it's highly specific. And if you've got samples that are really precious, you don't have much to go, the beauty of the probe system is it allows you to multiplex. So you can tag 1 probe to each target of interest, and you can look at somewhere between 2 targets up to 5 targets I've seen in your sample. So it allows you to maximize the amount of results or data that you can get from your sample. The drawback is that probes does cost a lot more. So for those of you who are new to qPCR, this video so I suggest that you visit this link, because this video, there's 2 videos in this link that shows you how the cybergreen assays and hydrolysis probe work. Now, once you decide on your chemistry, you do need to have quite a few considerations when designing your assay. So you do need to think about your target size. So 70 to 200 base pairs is generally the amplicon size that you work with qPCR. You want your GC content to be 50-60%. And if you're working with gene expression, you want to design your assays to span introns to avoid picking up genomic DNA contamination. With primer properties, you also want to think about maybe designing all your primers to have the same annealing temperature, so typically 60 degrees. So it allows you to analyze multiple targets on the same plate on the same run. You want to avoid GC clamps, avoid secondary structures, and also avoid primer dimer formation. If you're designing hydrolysis probe assay, you want your probes to be 5 to 10 degrees higher than your primers, your GC content to be 30 to 80%. You also want to avoid ending your probe design with a G because that acts as a natural quencher. So there's a lot to consider here. You can save some time by using a pre-designed assay, such as our Kickstart pre-designed cybergreen assays or probe assays, or you can pick a published primer probe design. So my suggestion here is whichever method you choose, always validate your qPCR assay. If you choose to design your own assay, we do have great tools that we can offer to you. They're free to use. So we do offer the Oligo Architect, so you can access them through this link here. So our Oligo Architect is a software that allows you to design both probe and cybergreen assays, as well as LNA designs as well. So we spit out a few designs which you can pick from. I suggest if it's the first time you're validating this assay, select a few designs and then pick the best one to save you time. And then you've got the Oligo Evaluator. So this allows you to check your sequence before you order them and run them on your qPCR. So this allows you to look at things like the melting temperature of your assay, check your assay for primer, dimers, as well as secondary structures. And if you require some assistance with resuspension and dilution guidelines, the Oligo Evaluator software will help you with that as well. Now we move on to qPCR assay validation. So you've got everything you need. You've got good nucleic acids, you've got your assay design. Now you need to consider validating your assay. When it comes to validating your assay, it doesn't matter whether you're starting with DNA or RNA, you need to take these steps to get good results. So you do need to validate your design using Oligo Evaluator. You also need to optimize your primer and probe concentration, as well as your reaction components and multiplex conditions. You also need to optimize your primer annealing temperature. You can do this by using the gradient function on your thermocycler. And finally, you do need to validate all your qPCR assays by running a standard curve, as well as performing a melt curve analysis for cybergreen assays. Due to the interest of time, we're not able to touch on every component here, but this is a great resource. So visit this link here, and it will take you through each and every of these components step by step and all the considerations that you need to set up your PCR assay. So what we're going to touch on here is the standard curve validation of your qPCR assay. So you need to set up a qPCR standard curve to determine the efficiency of your qPCR assay. In order to do this, you need to start off with a template. So it can be your cDNA, it can be spike plasmids or gene blocks. You then set up a 10-fold dilution of your template, starting from the most concentrated DNA sample. Each dilution should be run in triplicates. And based on your results, you will plot the CT values that you get from your qPCR standard curve against the known copy number. And then from this qPCR standard curve, you will derive this slope of the standard curve. You use this slope of the standard curve, plug it into this mathematical formula to calculate the efficiency of your qPCR assay. Now, you cannot always get 100%, but an efficiency of 90 to 110 is great, so it's acceptable. Anything outside of this range, you do need to go back and optimize your assay. So now, this takes us to the last poll, which is switch on. So how do you check if your cyber-green assays are specific? I mentioned this before, so if you're listening, you probably will get the answer right. I'll give you 10 seconds to answer this poll again. Okay, so let's review the answers to this poll. Hey, I'm so glad. A lot of you are listening. Perfect. So, to check the specificity of your qPCR primers, if you're doing cyber-green assays, you run a melt curve analysis or run it on an agarose gel post-analysis. So when you run a melt curve, what you do is post-PCR, your PCR machine can basically apply a gradual increase in temperature. So you've got your PCR products here, and as the temperature increases and as your PCR products reach their melt curve, they will separate and release the cyber-green in them. So your PCR machine will measure this fluorescence and spit out a melt curve that looks like this. So if you've got really nice and specific PCR products, you should get a single melt curve peak that looks like this. Now, if you're running your PCR assay the very first time, I also suggest that you run your products on an agarose gel and check that you get one single band on the agarose gel. Because sometimes a non-specific peak may be too small for you to pick it up on a PCR. You'll be able to see it on the agarose gel. Now, some other general tips that I'd like to impart to you today: make sure you run all samples in triplicates and that your triplicates should be within 0.2 to 0.5 CTs of each other. Make sure you pipette equal volumes of your DNA and validate all assays, including the ones you get from publication. Do not trust anyone but yourself. Now, important controls to include, because this will help you troubleshoot your assay. Make sure you use water as one of your negative controls, so always have an NTC. Always include your positive control. If you're running, if you're doing RNA analysis, include no RT controls as well. It's going to tell you if genomic DNA contamination is present. So you don't want that in your analysis. You can also try and reduce the risk by DNase treating your sample, your RNA sample, before you start your RT-qPCR. Now, when it comes to reference genes, there is no two reference genes or one reference gene that fit all experimental conditions. So I suggest don't just take primers or reference gene suggestions from your colleague or the lab next door. If you're working on a specific cell type but different treatment types, always validate. So always check that the reference genes is stable in your experiment. And I suggest that we also suggest that you pick at least 2 reference genes for normalization for good practice. Now, this takes us to the last bit of this presentation. So I've chosen 2 common qPCR issues to talk about. So the first common issue that a lot of people experience, or a lot of you experience, is there is poor or no signal in my qPCR assays. So these are the troubleshooting steps that I would take personally to troubleshoot my qPCR assays. So the first thing first is I'll look for my positive control. So I ask myself, did this work? If my positive control worked, it's telling me that my PCR assay is working fine and that perhaps the issue is with my sample. So you want to go back and check your sample purity and quality. You want to clean up your samples if they're not pure. If your RNA integrity is not great, then you might have to start from scratch. Now, if you've checked all that and it's still, you can't detect an issue, it could be possible that an inhibitor is present in your sample. So you can try and dilute these samples out to see if you can dilute the effects of your inhibitor and get your PCR to work. The other option is there are some master mix that are, so for example, the Kappa probe force that we have, these master mix are made to tolerate inhibitors a little bit better. So you can try these master mix as well. Now, what do you do if your PCR product, if your positive PCR control did not work? It indicates that there's an issue with your qPCR assay. So you need to go back to what we've talked about. So optimize your assay, buffer, thermal cycling conditions. And if none of these work, you might also think about using an optimized qPCR assay, or maybe just try a fresh batch of qPCR assay because your qPCR master mix might have gone off. The next common issue that a lot of people encounter is actually poor reproducibility in your assays. So the steps that you will take, so the first easiest, I guess most obvious thing is to check your pipettes. So are your pipettes calibrated? And if you're actually pipetting small amounts, like 1 to 2 microliters of cDNA, you might want to avoid that. Dilute your samples out and work with larger volumes such as 5 microliters might help. And if you're still not able to get good reproducibility in your assay in after you've fixed the pipetting issue, you might want to check your primer sets for possibility of presence of primer dimers. So you want to use things like the oligo evaluator software to check for complementary and secondary structures. You can try and reduce your primer concentration as well to see if you can remove this primer dimer formation. You also can perform melt curve analysis. You can use this new melt software that you can get on the website to check if there's melt, like a primer dimer being picked up in your assay. And if you can't fix this, you might just want to redesign your assay and start from scratch again, and that can help as well. So what we've talked about today is 3 main things I want you to take home. Sample prep, remember, garbage in, garbage out. So make sure you start off with good samples, QC your samples, check purity and integrity of your assays. Then we've also talked about qPCR chemistry and assay design considerations and the software that you can use to check your assay designs. And lastly, we finish off by talking about assay validation, so how you run your qPCR standard curve and determine the efficiency of your assay. So I hope you find this helpful. I've put together some resources, so you can go to these links. So this particular link here is really good for qPCR basics, but also troubleshooting. If you go into here, there is actually quite extensive troubleshooting manual. For those of you who are more advanced users, you might want to check this out. If you have extensive other issues, this might help you. This is the link to the UMelt software to predict melt curve for your sequence of interest. And you can also visit our molecular biology page and your PCR selection guide to help you pick the best mix for your PCR instrument. We will also share a PDF copy of this MyKey checklist. So this is like a summarized version of the MyKey guidelines. So you can go through step by step to make sure you've got everything you need for your qPCR analysis. And this is me. So this is my team. There's now 6 of us, so hopefully we'll have an updated photo of our team soon. But we're here to help you. We've also got a great technical support team. You can reach them at this number and this email address. If you go to the Sigma website, right at the bottom, you can find these links to contact our technical support and customer service team as well. This is my email address and my phone number. Feel free to add me, drop me a line on LinkedIn if you want to connect as well. I will now pass this session back to Yi Cheng and John to run the Q&A and conclude. Thank you for your time.
Thanks, Bernice. That was a wonderful presentation. And during your presentation, there was quite a few questions. John and I have been helping to answer most of them. There are a few outstanding questions. Which first one from an anonymous person is, they have a question about, in a disease setting, for example, risk allele or non-risk allele detection by SNP analysis, which is the best calibrator to use for qPCR? So I guess the person is asking if which control is best, the homozygous for the disease allele or homozygous for the non-disease allele, or heterozygous.
In a disease setting, which is the best calibrator to use for qPCR? That's a good question. I would think that you might have to set up both the homozygous and the homozygous and the wild type to be able to look at the difference in the peaks for your MELT curve. But if you don't mind sharing your email address, we could get in touch to help you out a bit more, because I think we need to understand exactly what you're doing there to be able to recommend the right controls.
I'll just, yeah, Bernice, that also might be a SNP genotyping.
Yep.
Exactly. Is qPCR, but it's not the gene expression part of qPCR. So yeah, you know, the disease state might be your wild type versus your homozygous, but it sounds like it might be a SNP genotyping event, which is a little bit different. Same concept, qPCR, but a little bit different.
Yeah, I think sometimes you use MELT curve analysis for these. So I would like to get in touch to be able to answer your question a little bit better, but I would imagine you probably need a wild type or a disease versus non-disease to be able to look at the difference.
Sure. Yeah, if you can, if that anonymous person can get in touch, that would be great. Another question is how to clean up the sample after examining the purity.
So you could, if you're using a column extraction method, depending on the kit that you have, check the user guide. Some column extraction methods allow you to run the samples through and wash them and clean out the impurities. If your column extraction doesn't allow that, you would do a 70%, or you would do a ethanol precipitation. So you can actually Google ethanol precipitation protocol for RNA or DNA. There should be some protocols, but if I've got your name there, I can send you a protocol that I used to use in the lab myself for cleaning up my samples.
Great. We have the latest two questions. How do I eliminate multiple peaks?
Okay, so that indicates to me that perhaps you may have non-specific assay, so a cybergreen assay. It could be, so sometimes it's actually a complex question. So sometimes when you have multiple peaks in your MELT curve, the main thing you should do is also run it on a gel to see if you run picking up multiple peaks on the gel. I would also check your primers if they're actually picking up, blast your primers to see if they're picking up other products. Check your primers, and also there could be contamination. So if you've got multiple peaks, make sure you might want to go back into your assay setup, start from scratch, make sure there's no contamination, use clean water. So that's the first step I would take to try and see if I can remove multiple peaks. Check your assay design, make sure it's not picking up non-specific products, because if there's non-specific products, then you would redesign your assay. Yeah, but then sometimes if you do get these multiple peaks in your, depending on what it looks, I do need to look at the picture of your assay, but sometimes you get multiple peaks due to secondary structures in your assay, and sometimes you cannot avoid it, but you can try and fix it. If you run it on an agarose gel and you only see a single band, you can add in some PCR additives like betaine or DMSO to try and help you get the best results from such an assay. But yeah, if you are currently experiencing this issue, get in touch. We'd like to try and help you.
Thanks, Bernice. In the interest of time, we're slightly over time now. I'll have to end the Q&A there, but we've got some more questions that we can follow up directly with the individuals. So I'll hand it over to John to close the webinar.
Thank you, Yi-Ching. Thank you, Bernice. Yes, for those outstanding questions, I can see names here, not anonymous, so we'll get back to you with those answers. If any answers that myself and E typed in throughout the webinar aren't clear, or you need more information, again, please reach out. You have our contact details. Reach out, and we'll endeavour to get your answers to you. On that note, if I just move to the next slide again, please. I want to thank Bernice today for giving us her qPCR 101 seminar. Hopefully all the attendees out there found it enlightening and helpful, and you're just a little bit more advanced in your knowledge now with qPCR. Ending that there, in 2 weeks' time, we'll have the Picky Blinders tips for improving LCMS sensitivity. I'll be surprised if any of you qPCR buffs out there will be attending an LCMS webinar, but please do. There's quite a bit to learn, or tell your friends who use LCMS. On that note, we thank you again once more. On your way out, if you could attend to the survey and fill it out, they'll be most welcome. And that's it from all of us. We bid you thank you once more and farewell. We'll see you again next time.